Wait just a second here to get the audio to click in. Should be good to go now though. So again, yesterday, we met in a team meeting, talked about kind of the week. You saw this. Today is the video lecture. I'm actually going to probably link this or kind of split this into two different links, just because it's a little bit longer, but then that way you kind of got a natural stopping point, and then you can kind of look at either watching it Tuesday or divide it between kind of Tuesday and Wednesday. Remember, Wednesday's a work day. Got those constant questions you can be working on, finishing the video lecture, reviewing for the quiz, or working on optional guided readings. Thursday, those optional guided readings, chapter 9 is 10 are due. If you do want to do one, both, or none, again, it's your option. And then Friday is the quiz on these notes that we're going through or starting right now. Constant questions are due on Friday, and then those optional chapter 16 guided readings or guided reading is due on Monday. Okay, so just some reminders for you. Let's kind of jump right into things and get going. So we're gonna be, talking prokaryotes and viruses today. Prokaryotes as a particular type of cell that lacks a nucleus. Viruses, they're unique. Obviously, you've heard a lot about viruses in the news lately. We're going to put some perspective on really truly what they are within this lecture, primarily in the second part or part two. To put some perspective on kind of where we're at, we looked at earlier this year when we looked at kind of biodiversity and the sort of the evolutionary tree, the sort of 5 kingdom system. You have the bacterial cells, which includes the eubacteria and archaebacteria, which would be kind of kingdom number 1. We have the protists here. That are, and these are all eukaryotic cells sort of above the line here, okay? And these are all prokaryotic cells below the line. We have protists, fungus, animals, and plants. So that's 2 or 3, 4, and 5, okay? Within this unit, the goal is to talk about sort of each one of these 5 different areas or 5 different kingdoms and take a bit of a survey on what's some major components of these 5 different kingdoms. And today we're focusing primarily down here on the eukarya and archaebacteria. You'll notice that viruses aren't anywhere to be seen on this sort of tree of life, if you will. And there's a reason why that is, because viruses sort of by rule really aren't truly a living thing. And they aren't truly alive. And we're going to talk a little bit about what that means. Basically, they're a type of mechanism that uses other living cells. Okay, so we'll get into that later on in part 2. So as far as prokaryotic diversity goes, so these 2 different sort of subsections of the prokaryotes. Remember, prokaryotes lack a nucleus, are the archaea and bacteria. Archaea are those that live in extreme environments, and archaea comes from the word archaic, and archaic means old. So these are things that live in really extreme environments, like the thermophiles live in extreme heat. You hear the word like thermometer. Halophiles refers to halite. Halite is salt. It's the mineral salt. So it can live in high salt concentrations. There's anaerobes. They're anaerobic, meaning that they can live in the absence of oxygen. Archaea comes from the word archaic. Some of the first cells on our planet, as for cells evolved that were prokaryotic cells, were those that lived in these extreme environments. And some of these, the cells that are related to them, are still in existence today. And the other branch of it are the bacteria, and these are kind of the typical bacterial cells that inhabit things like our throat, and it's on our hands, it's on our cell phones, and things like that. These are bacteria that live in our gut. They contain a peptidoglycan cell wall, which is unique to this domain. And then just sort of kind of want to say this out loud, from the cell wall, that it contains peptido, we've heard that, peptide bonds. So peptide bonds are those that are found in proteins, right? It matches up or bonds up amino acids. So you get a sense that it's a combination of proteins. And then glycan comes from glucose or glycogen. It's a carbohydrate. So the cell wall that is present within these bacterial cells is unique to this particular domain. Now, as we look at this, right, the typical bacteria, these are the ones that we kind of are in contact with or are probably more familiar with. Not to downplay sort of the importance of archaea, but they are a little bit less common, I guess, if you will, because obviously, we aren't necessarily experiencing some of these extreme environments. How do we sort of look at classifying prokaryotes in terms of the ways that we describe them? We look at kind of three big things. We look at cell shape, the type of cell wall that it possesses, and then its motility, or motility is a fancy word for movement. Okay. The cell shape-wise, there's three shapes. There's coci, bacilli, or spirillum. And to give you kind of a view of what I mean by that, so coci, these are spherical shaped. Bacilli, these are rod shaped, and then spirilla are types of bacterial cells they're literally spiral shaped. Okay. This gives you kind of an actual image of it, right? Cartoony sketches are great, but actual images. So these would be the coci, the spherical shape. If you hear, if you guys ever had strep throat, it comes from the bacterial cell called Streptococcus. These are round, spherical shaped cells. Bacilli, these are the rod shaped, and then spirillum, you can see these sort of spiral shaped cells. So first way that we kind of classify and define prokaryotes is according to their shape. Next way is we look at their cell wall, two classifications based on cell wall type, using this thing called Gram staining. So this is kind of cool. It uses a stain that if the stain adheres to the cell, that means it is considered gram positive. The stain, it actually uptakes the stain. And that's an indicator that it has just the peptidoglycan cell wall. And then gram negative, meaning that it doesn't uptake the stain. These are ones that have the cell wall plus an extra outer membrane. So looks a little different, like gram negative versus gram positive, right? So kind of jumping back and forth. Gram positive just has the peptidoglycan cell wall. Therefore, the stain is uptaken by the cells. Gram negative lacks that cell wall, or I'm sorry, gram negative has that extra outer membrane. So therefore, will prevent it from uptaking that stain. The stain adheres to the peptidoglycan. Now, to give another visual of that, here's a gram positive cell, bacterial cell. There's that cell membrane, phospholipid bilayer, and then there's the peptidoglycan cell wall on the outside of it. A gram negative cell there's the plasma membrane, there's the peptidoglycan cell wall, but then above it, there's this extra membrane, this extra phospholipid bilayer. So gram negative cells kind of have this sort of extra layer of protection in a sense. And that's partly function-wise, gram negative cells, and why do doctors, when they look at different bacterial infections and want to know, is it a gram negative or gram positive cell? Part of the reason is that those that have the gram negative, having that extra outer membrane, are often ones that are a little bit more robust, tough, if you will, which requires different treatments. They are a type of bacterial infection that can be harder to treat because of that extra outer membrane. And then finally, motility is another way that we classify prokaryotes. Flagelli, there might be one sort of long whip-like, or one or two, or many on a cell. So these are sort of the long, these whip-like appendages. This particular bacilli or rod-shaped bacterial cell has quite a pretty decent amount of flagella, and they act like this sort of whip-like tail in a sense that propels the cell. And then there's also these things called pili. These are very short and numerous. They're kind of like the sort of short hair-like structures that protrude off the edge of this, or the outer surface of the cell. And what a pili do is they're basically, think of them as cell attachment points. Um, it basically, bacterial cells live in these kind of funky places. I mean, they can exist just about anywhere. They exist on the surface of my mouse. They exist on your cell phone. They're inside our stomachs. They live in the back of your throat, inside your mouth, on your eyelids, on your hands. And what do they want to do? They want to stay in place, right? They need to be able to find a way to they've evolved a way to be able to stay in place by having these sort of finger-like projections off of their surface that helps them attach and stick to different surfaces. So they're certainly used to that. Okay. All right. Now, truly one of the big things that makes, you get the sense that like sometimes we think about prokaryotic cells as, yeah, they're simple and they lack a nucleus, and they're maybe less complex and all that stuff, right? But when we think about in the grand scheme of things, and from an evolutionary perspective, it's not about what's better and what's worse and who has the better body plan and things like that. Prokaryotes are quite successful at living in some pretty crazy places that would be unheard of for any other organism, especially multicellular organisms. So part of that, their ability to be as successful as they are and to be able to live as well as they do, if you will, is the fact that they have a short life cycle and they can reproduce in really short periods of time. So short life cycle, on average, bacterial cells will have a life cycle of about 20 minutes. And what's the significance of that? It just means that in about 12 hours time, you can go from a 1-cell bacterial infection to 68 billion cells. And if any of you guys have ever had any sort of bacterial infection, like say strep throat, I've had strep throat, and you feel fine. Maybe you get a little bit of a scratchy throat or something like that, and then all of a sudden you go to bed and you wake up the next morning and it's like, oh my gosh, your throat is sore, you've got a cough, it hurts, and you have a bacterial infection, you have strep throat. And within a relatively short period of time, you can go from a small number of cells to a lot of cells. And that's kind of truly one of the things, or one of the main reasons why bacterial are as successful as they are at being able to exist and sort of survive situations, is the fact that they can reproduce very quickly. So they use a process called binary fission. It's a form of asexual reproduction, which means that the daughter cells, when these cells divide, are genetically identical. It's simpler and faster than mitosis. It involves no chromosomes. There is simply less DNA. There's smaller amounts of DNA to have to duplicate and divide among the two cells and produce identical cells. And then the other thing is that DNA replication actually occurs simultaneously as the cell divides. Remember, in mitosis, we learn about, in mitosis or cell division, the DNA first has to duplicate, and then after that, the cell goes about dividing. In the case of prokaryotic reproduction, it's kind of all going on at the same time. So it is a bit of a time saver in a sense. Okay, so it produces identical cells. So it's just sort of an image of binary fission to give you sort of an idea. So there's that DNA in the nucleoid region. We don't have a nucleus. There's no membrane around it. DNA duplicates, and the cell division simultaneously happens while the cell is dividing and separating. So remember, still kind of this sort of inside out concept that we saw in plant cell division because of that presence of a cell wall, right? Now, you might go, okay, well, here's sort of a, when you think, it says, right, we just said this, produces identical cells, right? These things go through binary fission, which is a form of asexual reproduction. Okay. We go, well, then in theory, the bacterial cells that exist today would genetically be identical to the bacterial cells that say existed, 2 billion years ago or 100 million years ago, whatever. Okay. So the reality is, though, is that's not true. Okay. And the reason it's not true is that they do and have found ways to acquire genetic variation. Remember, we learned in kind of our last unit, and as we've learned in our unit on evolution earlier this year, genetic variation and variability within organisms is highly effective in being able to ensure that that species survives, right? The fact that there is a multitude of different traits and variabilities of traits, or alleles, if you will, that we learned last unit, is of benefit to living organisms, because then with changing environments through time, there is a possibility that certain segments of those populations of that species will ultimately survive. Some may not, but it ensures that that species still goes on. Well, prokaryotes sort of go about obtaining, if you will, genetic variation through some interesting methods, right? First is mutation. So the fact that they have a short life cycle creates a high rate of mutation. Well, yeah, I know they have a smaller amount of DNA, but the fact of the matter is that because they divide so quickly, okay, there is a chance that there are going to be mistakes made. And we talked about this in mitosis and meiosis. It happens sometimes, right? And when DNA is duplicated, you're talking about, an entire library full of books that has to be duplicated each time. And there's a chance that there's going to be mistakes that are made, and that certainly does happen. These resulting mutations can lead to different traits or characteristics, allowing these prokaryotes to survive better, right? Transformation is the absorption of small segments of DNA from the environment. So sometimes prokaryotic cells in the environment that they exist will come across DNA that they will absorb. By absorbing that DNA can then become basically, spliced into their existing DNA, creating a new and novel sequence or genetic variation. Another method is conjugation. Think of this really as bacterial sex, in a sense. Two bacteria connect temporarily and exchange DNA. So bacterial cells they'll connect between these pili. So these pili will overlap and allows for the exchange of genetic material between these adjacent cells during conjugation. And then finally, transduction. And this kind of somewhat bridges the gap in our discussion to viruses, but a viral infection, and really we're going to learn about this later, and we talked about this a little bit yesterday. Viruses, what are they? Basically, viruses are DNA packed in proteins for the most part. They are DNA injectors. So viruses will infect and inject certain sections of DNA into bacterial cells, thus changing them genetically. Okay, so transduction is sort of the fourth and final way. So here's just a few of these. So here's conjugation, those pili connecting between adjacent cells, allowing for the exchange of information. So there's, wrong screen. So there's the conjugation or bacterial sex. We have transduction. Remember, we just said transduction is a viral infection. So here's a little virus there occupying the surface of that cell, injecting its DNA into that cell that can become part of its genetic code. Here is also transformation. Sometimes, obviously, bacterial cells die as they break apart. They release their genetic material, and they may have certain gene sequences that are then absorbed by other bacterial cells and causing that cell to become more genetically variable. And a lot of times, this is certainly ways that we see different resistances being uptaken by other cells. This cell may have a resistant gene to, say, certain antibiotics, and that resistant gene that gets absorbed by another cell, and now that cell becomes resistant itself too. The antibiotic resistance is something that has kind of come to light in the last couple decades, especially because we used a lot of antibiotics for a lot of decades. And because these cells rapidly reproduce, we see mutations. Those that mutate and end up that have antibiotic resistance survive, and then we no longer can use certain antibiotic treatments. So we'll have some more conversation about that later, but I think it's good to say it at least now. Okay. All right. Onward. So prokaryotes, how do they obtain energy? What are their sort of modes of nutrition? So nutrition is how an organism obtains energy in carbon atoms, right? So energy in order for it to do the jobs it has to do, as well as carbon atoms to build up basically tissue and materials, right? Not tissues, cells and materials within those cells. So bacteria can be autotrophic or heterotrophic. We've heard those terms before. Autotrophs are those that use carbon dioxide as a carbon source. Heterotrophs these are autotrophic organisms like plants, right? We said before, they can use carbon dioxide and produce sort of their own food in a way. That is true within the prokaryotic domain, within prokaryotic cells like this. And then heterotrophs are those that have to consume organic molecules to use as a carbon source. Or heterotrophic organisms are consumers by definition. But there's also this is kind of cool, like in bacterial cells, there are also these things called chemotrophic and phototrophic organisms. They obtain energy from, chemotrophs will obtain energy from inorganic or organic compounds that, especially the ones that consume from inorganic compounds. These are non-carbon containing compounds. So, bacterial cells that can obtain energy by existing on the surface of a rock, right? That contains completely inorganic compounds. They have evolved a way to obtain energy from other resources or other sources. And then phototrophs, right, are those that would obtain energy from sunlight. Okay? So this is just kind of a little chart to kind of put the names to the different sort of energy source, either light or chemical, carbon source, either carbon dioxide or the consumption of organic molecules. So those that use light and carbon dioxide would be photoautotrophs. Those that use chemical resources and carbon dioxide are chemoautotrophs. Those that use light and consume organic compounds, these are photoheterotrophs. And then those that use chemical energy sources and use organic compounds as their carbon source are called chemoheterotrophs. Okay, so the name itself has meaning or significance in terms of how we sort of divide these different bacterial cells up in terms of their modes of nutrition. So we've looked at kind of how bacterial cells are classified according to how they there we go, their shape. We've looked at how their cell wall plays a role in how we classify and organize them, their motility, and then in addition, we've now looked at how their modes of nutrition. So, one of the perspective pieces that I want you guys to kind of pull from this is that, these small, tiny, quote unquote, insignificant, single-celled, simple prokaryotes, right, lacking a nucleus, are really quite diverse and really are able to occupy lots of different spaces and areas that one would never necessarily think that things could live there. And that's, I think, one of the kind of the cool things that you need to sort of think about. We take for granted sometimes those things that maybe aren't maybe as cool, and then, sometimes these are the creepy crawlies, right? These are the things that can make us sick, but there is obviously use for bacterial cells. Okay, so which gives us some thought right now, and that's part of the reason why we're transitioning to this slide. Prokaryotes play a role in our biosphere. First, by oxygenation of our planet. Earth's early atmosphere had very little oxygen. These things called cyanobacteria evolved, and these were the first photoautotrophs. So they were able to use light and carbon dioxide in our atmosphere, much like plants in a sense, and they begin to oxygenate our atmosphere. So these were the first organisms that began to sort of tip the balance of our atmosphere and slowly build up oxygen in our atmosphere to a level that it could support eventually and ultimately multicellular life. They play a role in chemical cycling. Nitrogen-fixing bacteria in the soil convert nitrogen gas into usable forms for plants. So these bacteria take nitrogen in the form of gas in our atmosphere that's unusable and converts it into a usable form that is in soils that is uptaken by plants. And that's how we obtain the nitrogen that we need in our bodies. There are heterotrophic bacteria that consume carbon and decaying organic materials. So they are decomposers, right? They play a role in the decomposition of material. So going back to our unit on ecology, they can be used in bioremediation. They play a role in bioremediation. They naturally will remove pollutants from the air, water, and soil. There's even, some pretty cool things that different companies have genetically engineered different types of prokaryotes to, like in the case of like oil spills, that they can release bacterial cells into the environment where that oil spill is taking place, and they will consume the spilled oil. So we can use even naturally occurring as well as even genetically engineered prokaryotes to play a role in basically cleaning up the environment in a lot of ways. Okay. And then last, to kind of give us some perspective, okay, is that bacteria, like we said, there's certainly some cool parts to them, right? They play some pretty significant roles. Prokaryotes play a role in the fact that we didn't necessarily say this, but they're in our gut, in our digestive system. We have healthy bacteria that play a role in aiding digestion. If we didn't have them in our gut, our digestive tract, we would have problems. But bacteria also are certainly known as what are called pathogens. Okay. And a pathogen is sort of, again, you guys have gotten used to this in biology. There's always these kind of fancy words. A pathogen is that which causes disease, right? So bacteria can often, and they do often, cause disease. So how do they do that? Basically, kind of three primary ways. They invade cells and will work their way into cells and destroy the cells that they invade. There's some pretty crazy types of bacterial infections. I'm going back to this slide, like that are spirillum based. Okay, I'm blanking on the word right now. I should have it. The sickness or the illness. I'll probably come up with it later, but it can inhabit like different tissues. It'll wind its way and literally sort of corkscrew its way into these tissues and invade these other cells. So there are different bacterial cells that will do that. They will secrete toxins that are harmful to the cells that they are maybe inhabiting or invading. And then the cell wall itself can be toxic. Some people have some pretty severe allergic reactions to some of the components of different cell walls, depending on if they're gram positive or gram negative. Now, what's our protection from pathogens? Okay, our skin is a protective barrier. Mucus within our sinuses and our digestive tract, the mucus that we have is basically a net in a sense. It catches bacterial cells that may invade our bodies. Our white blood cells are basically bacterial guards. They're on the lookout for invading bacteria, and they will search out, seek out, whatever, engulf and kill invading bacteria. And then we will treat bacterial infections with antibiotics. Antibiotics a lot of times are used they mess with the reproductive cycle of bacterial cells, which is kind of the biggest strong suit of what makes bacterial successful. Antibiotics there are chemical or compounds that will play a role in disrupting that. And then we also have different vaccines that can be used to treat and prevent different bacterial infections. Okay. And now beyond that, one of the things that we can protect ourselves from bacterial pathogens is simply good hygiene. So it's things like hand washing and covering your cough and cleaning up and disinfecting surfaces that are commonly used to prevent bacterial infections or bacterial pathogens. Okay. So this, and you're not meant to, you don't need to you don't need to memorize this or anything, but these are just some basic bacterial infections that some of them you're probably maybe familiar with. So different, pneumonia, otis media, or ear infections that I'm sure sinusitis. These are sinus infections. So streptococcus, upper respiratory. We talked about strep throat. Different types of food poisoning from things like salmonella, E. coli. You'll hear about that in the news where people will get if they don't cook meat properly or they don't properly wash materials before they consume them. E. coli and salmonella will cause food poisoning. Okay, so there's lots and lots of different types of bacterial infections that can affect our bodies. And again, bacterial infections can be treated with antibiotics. Okay. Now, we're going to stop there because we're going to end up going to part 2. I'm going to divide this up into 2 different links, or 2 parts. And we're going to find out about viruses in the next part. Viruses, part of the reason why viruses are harder to treat and deal with is the fact that antibiotics don't work on them, right? Viruses act and behave and are not truly a living thing. So that is part 2. So I'm going to stop right here. So this may be a good point for you guys maybe to pause or go take a break and come back, click on link 2, or save this for tomorrow and watch part 2 tomorrow. Okay. All right. We'll see you in a few minutes for part 2.